thin barrel is a folded protein structure that allow enzyme to generate geometrical active site at the 8 terminal ends of the parallel beta sheet ends. It also acts as a domain for the enzymes. The word T itself comes from topoisomerase enzyme, which is this structure is first found in topoisomerase enzyme. Besides that, thin barrel is also found in many enzymes which catalyzing totally different, unrelated reactions. But there are 15 distinct enzymes undergo this kind of framework.
Here. Yeah, down here. Come, have a seat. Okay, so side.
Thin barrel is consists of 8 alpha helix and 8 parallel beta strand. The structure name is after isomer triose phosphate isomerase, a conserved metabolic enzyme. So, thin barrel is one of the most common protein folds.
Thin barrel is considered as alpha and beta protein folds due to the alternating alpha helices and beta strands in a single domain. It forms a solenoid that curves around to close on itself in a donut shape, topologically known as toroid. So at the center here, it is tightly packed, mostly with bulky hydrophobic amino acid residues, but you will still need a little bit of glycine to allow movement. So at least 15 distinct enzyme families use this framework to generate an appropriate active site geometry.
Hello, my friend. I'm here to tell you guys about the thin barrel function.
Shut up! I'm playing games.
I'm sorry.
sorry. Never. I will continue from him. So don't mind him just now. Okay. So basically, I will continue with the functions of the thin barrel. So as we all know that this thin barrel is called as the triose phosphate isomerase, right? So it will work as a normal enzyme and it acts as a domain, right? So as we all know that domain has several functions like stabilizing, proofreading, as a binding of substrate. You don't come again lah. Yeah, so basically what the domain can do, this thin barrel also can do and act as a normal enzyme. Okay. Other than function as a domain, thin barrel has a diverse enzymatic reaction catalyzing 5 of the 6 classes of biochemical reaction. And one of the notable function is thin barrel can completely catalyze unrelated reaction.
Oh, hello, mate. I got another thing to tell you guys about the thin barrel, which is the loop region. So, listen carefully. There's approximately about 200 residues that can fully form a thin barrel. But, my friend, 160 residues are structurally equivalent between different proteins which sharing the protein folds. Then the C terminal ends tend to have the active site. And then those residues are required to maintain the structure. One last thing, my friend. Those loops can be so long that they contain much more other protein domains. That's all.
Looking for team barrel, you can actually use bioinformatic tools to predict your team barrel sequence. The first one is Team Finder and the second one is HSREP-ID. Let me show you.
Team Finder, a team barrel fall recognition system, is a software to predict whether your sequence belongs to team barrel or not. It is just simply by inserting your sequence and click submit. Another one is HSREP-ID, which have the same function as Team Finder.